For centuries, cannabis was used for its calming and medicinal effects, but no one really knew why it worked. In the 19th century, scientists tried to isolate its active ingredients, but with limited tools and a lot of guesswork, early researchers mistook nicotine and other harmless compounds for the cause of its effects. Some even tested cannabis extracts on themselves, leading to some wild and dangerous episodes. In 1896, researchers in Cambridge extracted a sticky red oil from cannabis. It showed narcotic effects, and from it they crystallized cannabinol, CBN, believed to be the plant's intoxicating principle. But it turned out to be inactive. Still, it was a step forward. By the 1940s, real progress came. American chemist Roger Adams, funded by the U.S. government, studied cannabis from industrial hemp, which ironically lacked THC. He identified CBD and synthesized variants like delta-8 THC and delta-9 THC. Across the Atlantic, Alexander Todd worked in war-torn England using hashish smuggled in diplomatic pouches. Both men, using different techniques, pushed cannabinoid chemistry forward. In Cold War-era Czechoslovakia, František Šantový nailed the structures of CBD and THC using just infrared spectroscopy and deduction, but his work remained obscure. It wasn't until 1964 that Israeli scientists Raphael Mechoulam and Yechiel Gayoni isolated and confirmed delta-9 THC as cannabis' intoxicating compound. This discovery finally gave science a clear target to study. By the 1970s, cannabis use had exploded globally, and researchers began testing pure THC on animals and humans. They found nearly all of the plant's psychoactive effects came from THC, but how exactly it worked in the brain was still unknown. At first, scientists thought THC just melted into brain cells due to its oily nature. That theory changed in the 1980s, when Dr. Alan Howlett discovered that THC binds to a specific receptor in the brain. This was the CB1 receptor, now known as the first identified cannabinoid receptor. In 1993, a second receptor, CB2, was found, mostly on immune cells. The big question became, why would our bodies have receptors for cannabis? The answer came in 1992, when Mechoulam and Bill Devane discovered anandamide, the first endocannabinoid, a molecule our body produces naturally that binds to the same receptors as THC. Soon after came 2-AG, another natural cannabinoid. This launched the discovery of the Endocannabinoid System, ECS, a network of receptors, molecules, and enzymes that regulate balance in key functions like pain, mood, sleep, appetite, memory, and immune response. What started as a mystery about a plant turned into one of the most important biological discoveries of our time. The ECS isn't just about cannabis. It's a built-in system in the human body that helps maintain balance.